When a substance is exposed to laser light, nearly all of the scattered light produced is Rayleigh scattered light. However, a very small percentage is Raman scattered light. Because the percentage is so small, this light is invisible to the naked eye. However, with the use of certain tools, this light becomes visible. This is the spectrum of scattered light from the sample. Because we are using a 532 nanometer excitation wavelength, we can detect both strong Rayleigh scattered light at 532 nanometers and weak Raman scattered light at a longer wavelength. Light at 532 nanometers is visible as green light. Therefore, at first, a strong green light was observed. Raman scattered light, on the other hand, is too weak to be seen. Next, by using the filter, Rayleigh scattered light at 532 nanometers is blocked. Now, the Raman scattered light emitted at a longer wavelength is visible. This is the Raman spectrum of water. When using an excitation wavelength of 532 nanometers, the Raman peak of water appears around 650 nanometers, the wavelength of the color red. That is why the Raman light was visible as red light. Usually, the horizontal axis of a Raman spectrum is indicated by wave number, not wavelength. By using wave number, we see the Raman spectrum of water independent of the excitation wavelength.
The sample is the aspirin in powder on a microscope slide. The microscope slide is placed on the motorized stage. First step is to visualize the surface of the sample. So, I will start LabSpec 6 software and I will select the video mode. For the sample visualization, I will choose 10x objective. Using the joystick and the stage wheel, I will focus on the sample, checking in the same time on the computer screen. As you can see, there are several crystals with different sizes and shapes, and I will select one of them. For the measurement, we will use a long working distance objective, for example 50x objective. Then we will adjust the focus on the sample again. This is the Raman spectrum of the aspirin obtained in 0.5 seconds. The Raman bands are narrow and very well resolved. Nevertheless, we note a small fluorescence background. It can be successfully removed by using an option called flat correction. So I will activate this option and I will launch again a real-time detection to see the changes. It can be easily observed that the background is almost eliminated. Now we have a very nice baseline. I will stop now the real-time detection and I will launch a Raman spectrum acquisition for 1 second. 2 accumulations will be used for a better signal-to-noise ratio. This is the Raman spectrum of the aspirin easily obtained in one shot, with a good signal-to-noise ratio, as you can see. For the further spectral analysis, we can follow, for example, the band maximas.